Hello, everyone. Thank you so much for tuning in today. My name is Angelique White. I am at the University of Hawaii, and I want to talk today about utilization of in situ in water optical measurements for hypothesis testing, primarily in the open ocean. I'm going to share a few excerpts of my own work, and then I'll talk a little bit about the challenges that remain in exploiting the optical properties of bulk seawaters as well as single particles. First, I really want to acknowledge the funding sources for the work that I'll share with you today: the National Science Foundation, the Simons Collaboration on Ocean Processes and Ecology, and Access to the Sea, and much, much more from the Hawaiian Ocean Time series. You'll hear a good bit of work from Matildas Langine, my collaborator Lori Jaranek, Fernanda Hendricks Freitas, the gang of Scope Gradients, and many, many others, as well as some tales of a few microbes. So I want to start in the beginning. One could argue that ocean optics is one of the oldest branches of oceanography, beginning with the advent of the Secchi disk. And around the time of the Challenger expeditions, the early thinking was that the color of the ocean was simply the reflection of the blue of the sky. And here's a commentary from a 1910 issue of Nature that politely tears that argument to shreds. And the author is musing on a lecture that they had recently heard from Lord Raleigh, describing how absorption and scattering of light leads to our own visualization of color. And the commentary ends in this highlighted section by stating that a visit to the screw well never failed to convince the doubter that the water contained particles sufficient to account for the perceived color. The diatoms got all the attention in this. They'll get less attention in this talk in particular. So we've come a long way. There's been an explosion of optical oceanography, particularly in the last few decades, with remote sensing growing up from multispectral to hyperspectral, deployment of an army of bioargo floats, the advent of full ocean depth moorings, profiling wire walkers, gliders, AUVs, armed to the teeth with sensors and cameras. So now when we think about what we learn from ocean color, we're talking about energy fluxes, scattering, and what that can tell you about particle concentrations, organic and inorganic carbon, absorption, and what that tells us about pigment diversity, fluxes of organic matter, diavertical migration even, particle size, community composition, and a whole array of approaches aimed at understanding ocean metabolism. And that's what we learn from this, beautiful range, spectrum of ocean color. This is an image from Marcel Fernard, and I have added on the beautiful blues of the Pacific as the hot. So I'll give you just an outline in vignettes of where I'm going to go from here. I'm going to talk about some work that we've done asking who's there from a plankton perspective. And most importantly, what are the environmental conditions that drive changes in community composition? This will highlight some work from, I'm sorry for the acronyms, the LIST, which uses forward scatter to estimate particle size distributions, living and dead particles, the imaging flow cytobot as well, that images individual particles in slightly different size ranges. Then we'll talk about something we don't talk about as much, how do they die? And I'll show some highlights from various network modeling work and really the rich amount of information we can get by combining these various approaches. And last, I'll end with some thoughts about future efforts, as this community is really faced with what is a large scale data integration task across various platforms. And hopefully that'll come through in some of these vignettes. Okay, so I'm going to start with some work from Lori Jaranek, myself, and others that was recently published in 2020 in Global Biogeochemical Cycles. We really were talking about the importance of this phytoplankton middle class to oceanic net community production. On the bottom here are some of the constituents of the middle class. They're quite beautiful and diverse. So in framing this work, we were really focused on these conceptual models of the particle size distribution that are so canonical in our literature as drivers of nutrient supply. This is the classic bifurcation model of Legendre and Levave from 1989, where you move from this oligotrophic regime on the far right, where small cells are effectively drawing nutrients down to incredibly low concentrations. They dominate the phytoplankton community structure. The microbial loop and remineralization, sort of refeeding of those organic materials back to the phytoplankton community, dominates these fluxes. And then the sinking flux of particles out of these ecosystems are presumed to be driven by aggregation and slow sinking rates. As you increase nutrients, you move to the left and you start to be able to support large phytoplankton. And in this case, within this model, the sinking of these organisms are driven by direct sinking flux of ballasted phytoplankton cells, as well as grazing of those cells by larger grazers and the production of fecal pellets. So in this sort of canonical sense, there are very different pathways towards producing exported material. And it's fundamentally driven by nutrient supply and a shift, a bifurcation in the particle size distribution from small to large. So this is what we sort of started with as how we thought this system works. And our hypothesis was really that these changes in particle size distribution across ecological gradients are predictable, and they are based on resource supply rates and ratios, and that will drive patterns, observed patterns in productivity and export. Here's a little cartoon that I made to try to illustrate that. As we move from the oligotrophic subtropics in the North Pacific, we move from an area dominated by small cells, low primary productivity, and low rates of particle export. As you move into the transition zone, where you start to see uplift of isopycnals, intrusion of nutrients into the euphotic zone, the particle sizes become larger, productivity increases, until finally you're in the rich subarctic, where there are residual nutrients, some evidence of iron limitation, and larger organisms dominating phytoplankton community structure. And across these gradients, we expect to see variability in productivity, variability to export, variability in nutrient fluxes. So this was, for us, the ideal setting to test this bifurcation model. And this is what we've done. These were 3 transects, so went back a couple times, 2016, 2017, 2019. These are all along 158 west from Hawaii to roughly 40 north. And you can see we transited across about a 15-degree range of temperature. The red is the physical front, so a salinity front, and the black is a chlorophyll front, so a biology front. As you move from the subtropics, a low nutrient environment, this is nitrate, across that chlorophyll front, you start to see increasing nitrate, increasing phosphate concentrations, increasing concentrations of particles that lay down on a GFF filter. This is particulate organic carbon, and a very large 2- to 3-fold increase in net community production based on oxygen argon. All right, so this is a perfect transect to ask questions about particle size. Now, when we started analyzing the data, it was a little different than we had expected, right? There were statistically significant relationships between the middle class, these nanoplankton, 2 to 20 microns in a spherical diameter, and the rate of net community production. And these were significant year to year. And so I'm showing you here on this left plot, the concentration of biovolume of nanoplankton in red in 2017 and in black in 2019, and the relationship to net community production. Right, these are very strong relationships. In 2019, we had the imaging flow cytobot, which is a slightly different way of approaching particle size. and we saw similarly strong relationships. You can look at these in time in 2017 as we moved to the north and then turn around and head to the south. 2019, we took a few crossings of the gradients, and you can see clearly that there are strong relationships between net community production in red. These are bottle measurements in the yellow, and the concentration of this middle class. This was not the bifurcation that we expected. This is definitely a non-binary result. So it was clear to us that the middle class is really thriving in this productive transition zone. These are what I call joy division plots. Some of you will get that, where we're looking at the concentration of particles in various size fraction here with traditional flow cytometry. This is an instrument called the C-flow. So looking at sort of pro and sen, the imaging flow cytobot, and the list. You can see in all cases, you see increases in biomass across that transition zone, both in the C-flow, the IFCB, as well as the list. But what I want you to notice here is in these smaller particles, you can see the sort of grow-in of the pico eukaryotes and Synechococcus. But when we look into the nanoplankton, from the subtropics into the upper edge of the transition zone, the mode of the particle size distribution is nanoplankton. You see a spreading of these distributions, but still nanoplankton are really driving these populations from the south to the north. Matildas Langine had done a wonderful job of breaking apart these imaging flow cytometry data and looking at the relative contribution of diatoms, for example, in green, dinoflagellates in orange, other dinoflagellates in pink. And what we recognized here is that at least in these 2019 crossings, these gymnodentid dinoflagellates really dominated that productive transition zone, both in biomass and in abundance. About 35% of the estimated nanoflagellate carbon was comprised of these often mixotrophic dinoflagellates. So again, this was an unexpected result that is a bit opposite of that sort of canonical bifurcation model. Now, it's important to place your work within the context of the greater canon of literature that's out there, and that's what I've tried to do here, just showing you using the Connected Papers app where our 2020 paper fits in the prior literature, as well as literature that has come out about the same time. And they fall in a few nodes, a whole range of papers looking at metabolism across ecological gradients, some papers trying to understand the transition zone itself, and this one really beautiful paper by Manoa in 2015, where they make the comment that biomass-specific production and growth rates are similar, and this was in cultured laboratory isolates, are similar in both small and large cells, but peak at these intermediate sizes. And the mechanism for that is that the ability of these intermediate sized organisms to exploit nutrients is high. And that explains their biomass dominance during blooms. So this is really this sort of lab, nature divide where we're seeing, all of these lab-based studies try to explain what we actually observed in the field. We're not the only ones thinking about a non-binary approach to production and export. Tammy Richardson has a beautiful annual review of the mechanisms and pathways through which small phytoplankton are exported and the role of smaller and middle-sized plankton in food webs. And Ben Ward and others have done a beautiful amount of work looking at size dependence of phytoplankton growth rates and trade-offs between nutrient uptake and metabolism. And that's what I've shown here. You can see the predominance of this 2 to 20 nanoplankton size range. All right, so it all starts to come together. And again, this was not the result we expected, but by combining an array of optical instrumentations, we're starting to evaluate hypotheses in, I think, new and powerful ways. So I'm going to shift gears a little bit and talk about some work from Matildas Langine that came out in Limnology and Oceanography this year. but the life and death of this nitrogen fixing organism called Crocosphaera. This is at Station Aloha in the North Pacific, and really was an effort to examine the fine scale predator-prey dynamics in this region. And so the hypothesis that we were working with was that the growth and loss of these dominant organisms in the North Pacific are tightly coupled. And this is built on years of measurements of optically based particulate organic carbon from transmissometers, ACSs, other instruments, where we see this daily rise and fall of carbon. All right, growth and loss is the way we've interpreted this. Although if you look at cell counts, they're relatively invariant, suggesting these tight couplings. So I'll kind of get to why Crocosphaera in a minute, but we hypothesized that Crocosphaera should be very effectively grazed and that the grazers would be specific nanoplankton protists, ideally in the 10 to 20 micron size range. And that's based on just the size range of Crocosphaera itself. It's about 5 microns in diameter. This is the large size fraction of Crocosphaera. So this is what we did to address that question. Pretty simple. All you got to do is classify millions of cells from the imaging flow cytobot. Matilda has developed about 200 classifiers for nanoplankton in the Pacific. Then use a cell size or cell cycle model to calculate growth of the main player, in this case, Crocosphaera. And you can see here, you can see singlets of Crocosphaera beginning to divide and eventually dividing into a daughter cell. We then looked for interactions. We built interaction models between the prey item, Crocosphaera, and potential predators. And in this case, predators, Dinophysis, Protoparadinium, Strombodinium, we were able to see Crocosphaera-like particles within their food vacuoles that really gave us confidence that our interaction models were telling us what we thought it was. Then you build a predator-prey model, in this case, Lotka-Volterra, and you can start to examine the time variant rate of change and estimate the timing of growth and grazing of these organisms. And I'm being a little sarcastic. It's not 4 steps. Each one of these is, I think, a little bit of a tour de force. I'm really impressed by our work. And so what we found out from this work was that at least at this time, the diazotroph Crocosphaera was about 30% of the nanoplankton biomass, a very dominant player in the nanoplankton. We could estimate growth rates of about 0.8 per day. It's growing pretty fast and near what you would see in culture. You can look at the timing of growth rates over the day. We saw a lot of division near dusk and then respiration over the nighttime period, as well as grazing losses. We were able to identify the heterotrophic Protoparadinium as the predominant grazer. Rates for about 0.6 plus or minus 0.4 per day. And you could see the timing between Crocosphaera on the top and the various grazers here on the bottom, with growth of the prey item, rapid consumption and growth of the grazer at night, and then a decline. And so this is, in my mind at least, a really novel demonstration of the tight coupling between a very specific predator and prey that's different than some of the bulk analyses that we typically are able to do in optical oceanography. So kind of going back to that sort of how this work fits in the greater canon, if you look at the Langine paper 2020, it's closely related to a little nest of papers trying to understand nitrogen fixation by this organism, a group of papers trying to understand diel cycles in general, and a whole web of papers really focused on understanding picoplankton, as well as this really beautiful paper by Sarah Hu, A Hard Day's Night. It's a great title, where they were able to show that most of the protistan groups in this area were characterized by phagotrophic taxa that had very high metabolic activities at night. There was a similar paper, Connell, that also found that heterotrophic nanoflagellates contained the highest ingested picoplankton prey at night. So these are both very consistent with our imaging-based results in terms of the timing of predation on Crocosphaera. So I would just say that with cooperation and rapidly developing technology, we can now assay whole planktonic communities in really unprecedented ways, starting to bridge omics and cell-based optical measurements all the way up to bulk optical approaches. So it's an exciting time to be in oceanography. There's also a really amazing expanding power of single cell imaging. This is from a paper in Frontiers, Lombard in 2019. And traditionally we focus on our preferred taxonomic group or a functional range that's specific to the instruments that we're able to afford. Models do the same thing. They simplify taxonomic diversity. Yet with this incredible developing range of technology, we're now able to see entire planktonic communities in really unprecedented ways, along with characterizing their environment, incorporating ecosystem modeling in our design, and ideally better understanding both bottom-up and top-down control processes. And I suspect these tiered instrumentation, collaborative approaches are really going to start to transform our understanding of ecosystem processes. So I wanted to end with a few conclusions and parting thoughts. And one, I'll just say that I hope what I've shared today makes it clear that an optical approach to oceanography using optical tools is really beautifully poised to move beyond creating products, but blend our understanding, our way of seeing the oceanographic world with omic approaches, with classic biogeochemical approaches, to test fundamental hypotheses of how ecosystems are structured and function, and to iteratively improve our global models. And so another thing to keep in mind is that no approach is perfect. I think we need to be very mindful of the limitations of our instrumentation and our approaches and all the ways that our data can deceive us. So know your instrument and test your assumptions. And last, and we'll hear this in other talks, I think this rapid expansion of platforms from floats to gliders and moorings will optimally incorporate more than just transmissometers and scattering and fluorescence, and sort of bring me back to that first bullet point of the power of cooperation in science. I'll end with a parting thought. This is past year's quadrant. Probably many of you have seen this before. On the X, you move from low to high in terms of applied science or consideration for use. You can think of someone that's very high, and that is the Edison for applied research. And then on the Y is sort of basic research, where you have a quest for fundamental understanding, curiosity-driven work. You could think of something like, Niels Bohr. Put this in NSF parlance, as you'd have broader impacts on the X and intellectual merit on the Y. And I'll go ahead and replace these guys with a few optical oceanographers, women in this case. I can think of someone like Shirley Jeffrey, known as the mother of chlorophyll, a good example of an optical oceanographer of doing basic research. The Clarice Jentsch, co-founder of Bigelow, and have done an enormous amount of work with the early ages of optics and applied research. And we'll let Mary Jane Perry be our passeur with use-inspired basic research. So I think that optical oceanography really can bridge the curiosity-driven all the way to the applied. You don't have to choose. So with that, I say mahalo to you all, and thank you for your time. And I look forward to the discussion when we finally meet for the plenary. All right, I'm going to stop recording.